Our topic today, guys, is the cell cycle. So as you guys have seen, we're kind of diving into each organelle, and today we're inside the nucleus. All right, so let's start with cellular reproduction. First, life begins with a fertilized egg. That's called a zygote, and that zygote goes through cell division over and over again. Cell division through mitosis gives rise to many identical cells, right? That means that something must be happening in order for cells to look different along the way, right? That process is known as differentiation. So differentiation is a process that creates special structures and function. During the process of differentiation, only specific parts of the DNA are turned on, right? Because you have to remember that all cells have the same DNA. All cells coming from the same person have the same DNA. So how is it that it becomes different, right? It just means that different parts of the DNA are turned on within specific cells, and that's why different cells look different and behave in a different way, right? All right, so stem cells are undifferentiated cells that can become differentiated into one or more types of specialized cell. Um, there are two types. There is embryonic and adult stem cells. Embryonic stem cells are cells that have never been differentiated, and they could become any type of cell. So they're super valuable. But adult stem cells are found in adult bone marrow, and they're partially differentiated, so they can only become certain types of cells. Once a cell is differentiated, that's it. Okay, once you've decided to be a muscle cell, you are a muscle cell for life. The cell cycle. The cell cycle is a repeated pattern of growth, DNA duplication, and cell division, right, that occurs in eukaryotic cells. So, three parts: growth, DNA duplication, and cell division. Main purpose of the cell cycle is to make new cells or to repair cells that have been damaged. Organisms make new cells instead of having their cells grow larger, right? Because if the cell grew too large, then at some point it wouldn't be able to do the processes that we talked about, osmosis, diffusion, and all that, to meet the needs of the cell, right? So cells will divide or reproduce before they become too large to function properly. So look at this picture. What I'm trying to show you here is the difference in surface area, right? Proportionally, the smaller cell has more surface area exposed than the larger cell. All right, some essential vocabulary. Chromosomes is a long, continuous thread of DNA that consists of thousands of genes and regulatory information. So when it's unwound in that way. Chromatin, right? Chromatin is a DNA that's loose and uncoiled when the cell is not dividing. So when the cell is not dividing, the DNA appears as chromatin. When it is, it appears as chromosomes. So chromosomes is when it's more wound up. Chromatin is when it's more unwounded. A gene is a section of DNA that contains the instructions for making a protein, right? And I want you guys to remember that we've been talking about proteins since macromolecules. And I believe I've said to you that arguably, they're the most, let me not say important, they're the most diverse. They're the most diverse macromolecules. A chromatid or a sister chromatid is one half of a duplicated chromosome, right? So one half of this, so this is a duplicated chromosome. A half of that is a sister chromatid, right? So let's finally get into the cell cycle. It consists of three main phases: interphase, mitosis, and cytokinesis. As I said before, interphase is when the cell grows. Mitosis is um, when nuclear division actually happens, and I'll fix that definition. And cytokinesis is the separation of the cytoplasm at the end. So technically, if a cell goes through interphase and mitosis but doesn't go through cytokinesis, you're still just going to end up with one big fat cell with two nuclei, right? So mitosis is the division of the nucleus, but cytokinesis is the actual separation of the cytoplasm. So interphase. Interphase has three phases. It has a growth phase or G1, an S phase, and a G2, a second growth phase. So G1 is when the cell grows and makes proteins. This is when the most growth occurs, right? Um, The S phase, which is a synthesis phase, is when DNA replication occurs. So this is when the doubling of the chromosomes occur. So in um, G2 is when there's more cell growth and protein synthesis. So at the end of interphase, the cell has two complete sets of chromosomes, two full sets. Everything is duplicated. The S phase, because of the S phase at the end of interphase, the cell has two full sets of chromosomes. After DNA replicates, other organelles also copy, right? So it's not just the DNA that's duplicating, the other organelles are duplicating as well, so that the cell can have those, right? We're just focusing on, excuse me, we're just focusing on the DNA. So then we have actual mitosis, and mitosis happens in four steps. I like to call those PMAT: P for prophase, M for metaphase, A for anaphase, and T for telophase. So prophase is when the chromatin becomes chromosome. So it's not just like a ball of thread anymore. It becomes condensed and it's visible as chromosomes. The nuclear membrane also breaks down, as you can see here. So the nuclear membrane is breaking down. So what does this mean? This means that, hey, maybe, excuse me, think about what that means. I'm not going to give it away. Centrioles separate and spindle fibers form between them. So remember back when we talked about centrioles being an organelle. Then we have metaphase, the M of PMAT. Metaphase, chromosomes line up along the middle of the equator or the cell. So M, middle. I think it's the easiest stage to identify because everything is in the middle and the name of the phase starts with M. Then we have anaphase. This the chromatin moves towards the opposite poles, right? And we have telophase or telophase. Spindle fibers break down and disappear. Chromosomes uncoil into chromatin, and two nuclear envelopes form around the chromatin. Then we have cytokinesis, which is the division of the cytoplasm. So let me just go back real quick. Notice that this is still one cell, right? But this process makes it two cells. So the cytoplasm divides and it forms two identical cells. In plants, a cell plate forms midway. A cell plate forms midway between the divided nuclei and gradually develops into a membrane. So in plants, it takes a little bit longer because of the development of this cell plate. In animals, it forms what's called a cleavage whorl that pinches in the cell into two equal parts. So a difference there. How often do cells divide? They divide based on their need, right? Hair cells are some of the fastest dividing cells, right? And you'll see why in when you do your next activity. The internal lining of your intestines every 5 days, approximately. Skin cells every 2 weeks, red blood cells for months, and liver cells up to a year. So just some FYI. The cell cycle is controlled and regulated. It starts and stops, and proteins are very important in this process. Goal of the checkpoints is to make sure that the cell is healthy and strong, completing each step correctly. Um, DNA controls these checkpoints. Mutation in the DNA that controls the cell cycle can lead to cancer. So if there is DNA that tells the cell cycle when to start, when to stop, and there's a mutation in here, then you can see how that could cause some serious problems for the cell. Ultimately, the cell cycle continues when it does not need to, and the cells keep dividing, and they eventually form cancer. Cancer cells divide much more frequently than regular cells do because they're not being controlled. So the checkpoints, again, they're important points that regulate the cell cycle. And there's pretty much one after every stage of the cell cycle. So there's one at the end of G1, and this makes sure that, everything has grown the way it was supposed to. Then there's one at the end of S, make sure that the DNA is synthesized in the way that it should, and basically, they just check at every point before they move on to the next, as you can see in that picture. Draw the picture. Apoptosis is programmed cell death. This is when there are internal or external signals that activate the genes that produce self-destructive enzymes. So the nucleus shrinks and breaks apart, and that's what happens when you have webbed fingers or webbed toes. Tumors, we mentioned them a little bit. They're just when cells grow out of control. There's two types. It could be benign or malignant. Benign is abnormal cells. They're typically clustered. They may be harmless and they can be easily removed. Malignant is the one you don't want to hear. They're cells that break away from the tumor and move to other parts of the body and can produce more tumors. And metastasis, or when a tumor metastasizes, is when it spreads to other organs. Causes of cancer. Cancer can be caused from many things, mostly biological factors. You are more likely to get cancer if you're predisposed to getting it genetically, right? Lifestyle choices can also play a role. Viruses and other infections can lead to cancer, for example, HPV, and then exposure to carcinogens, so exposure to cancer-causing agents, such as tobacco, smoke, asbestos. All right, that's it. You can see our next topic. Take your notes.